Hello friends, welcome to my channel Easy Life Science. Here we are discussing the Griffith experiment and how the DNA is discovered as a genetic material in every living cell. So subscribe my channel and share my videos. Let's start. The DNA is a deoxyribonucleic acid. At time for process of a transcription, the DNA get converted into the single-stranded RNA or a mRNA. The process is called as a transcription. Further, the genetic information stored in a RNA is converted into a chain of amino acid, that is a protein. The process is called as a translation. And this conversion of DNA to RNA and RNA to protein, scientists gave a name it as a central dogma of molecular biology. DNA to RNA transcription and RNA to protein translation, so that the genotype get converted into a phenotype or a particular gene get expressed. This is called as a central dogma of a biology. How do we know that all of our genetic information comes from a DNA? Or what type of experiment would you design to determine that the DNA is a source of all genetic information? Number of scientists are working on it, and it was concluded or it was proved after a Griffith experiment. The scientists Griffith were working on a pneumonia. The causative agent for a pneumonia is a Streptococcus pneumoniae. It is a one bacterium, and this strain causes a pneumonia to the animals. The symptoms like septicemia, fever, and death. These are symptoms of the pneumonia. And the scientist Griffith will make a use of two strains of pneumonia, that is of Streptococcus pneumoniae smooth strain and Streptococcus pneumoniae rough strain. Frederick Griffith was a bacteriologist who studying on pneumonia. He discovered two type of bacteria. One, that is the bacteria which produces a smooth colony on a culture media. That strain were named as Streptococcus pneumoniae S strain, and this S strain is virulent because the smoothness of a colony is because of the polysaccharide like capsule around them, and this capsule shows a capsular antigens, and so this strain or a S strain, S stand for a smooth, this S strain is virulent. Virulent means disease causing. It causes a pneumonia to the animal. The second strain was a rough strain or a R strain, and these organisms or these strain are not disease causing or non-virulent because they are without any kind of capsule or a polysaccharide. So no capsule. The colony of these strain are rough, smooth colony and a rough colony, S strain and a R strain. Smooth colony R or a smooth strain are virulent, disease causing, causes a killing of an animal, whereas the rough colony are non-virulent, non-capsulated, non-disease causing. The scientist Griffith made a use of these two strains, S strain and R strain. First step of a Griffith experiment, he made a use of a lab animal, that is a mice, and in a first step he injected a smooth strain of a pneumonia to the mice, and because of an injection of a smooth strain, to the mice get died, and because of the injection of a rough strain, mice does not get died, they remain live. Here you can see for a dead image of a mice in a left of a screen. This is because of an injection of a smooth strain. Smooth strain is a virulent, disease causing, and a fatal, or it is deadly. Further toward the right, injection of a rough strain, the mice does not get killed. Conclusion: the smooth colony or a smooth strain must carry a disease, or these are deadly. Then in third step, the scientist Griffith heat killed that deadly smooth strain and made an injection to the mice. And after injection to the mice, the mouse lived. Because of a heat killing, the antigenic property or a production of a capsule get loosed. So this heat killing denatures a virulency of the smooth strain. So after injection of such strain to the mice does not get killed, they remain live. This was a third step in his experiment. Then with the fourth step, the scientist Griffith injected the heat killed smooth strain and a rough strain. The mixture of these two strains were injected to the mice. As both of these strains in a mixture are non-virulent, heat killed smooth as well as a normal rough, these two strains are non-virulent, not fatal, not deadly. So after injection of mixture of these two strains to the mice, surprisingly the mice get killed. It was a surprising result. As we know, both the strains, heat killed a smooth and a wild rough, both of them are non-virulent, even though the injection of this causes a killing of a mice. It was a surprising result for a Griffith. And he was studying or working on the fourth step. Somewhat was there, because of which the heat killed a smooth strain gets transformed into deadly type. Somewhat was there, or a transforming principle was there, because of which a heat killed a smooth strain become deadly. And that transformed principle or a particle were studied by a Griffith, and the Griffith experiment did not prove that DNA was responsible for a transformation. How do you design an experiment to prove that DNA was responsible for a transformation? Further, the three scientists Avery, McCarty and McLeod, repeated a Griffith experiment, and they worked on a fourth step. I once again repeat, a fourth step was a mixture of heat kill a smooth strain plus wild rough strain. This mixture is injected to mice, and mice get died. And this fourth step, three scientists were worked heat killed a smooth strain and a rough strain. This mixture is added with enzymes or different kinds of enzymes. He just made 5 test tubes, and each test tube he added a mixture of heat killed the smooth strain and a rough strain, plus he added different kind of enzymes, and the enzymes which destroyed a carbohydrate, lipid, protein, RNA and DNA respectively. Here in image we can see for 5 test tubes, and each test tube he added the mixture of 2 strains, first, second, third and fourth, fifth. The different enzymes were used by these 3 scientists. The enzyme which denatures a carbohydrate, that is a saccharolytic enzymes, the enzyme which denatures a lipid, that is a lipase enzyme, the enzyme which denatures a protein, that is a proteolytic enzyme or a protease, and the enzyme which denatures the RNA, that is a RNase enzyme, and the enzyme which denatures a DNA, that is a DNase enzyme. These are the enzymes which denatures a particular component like carbohydrate, lipids, proteins, RNA and DNA. In each tube he added the mixture of 2 strains, and they was working on a 4 step how the mice get died. And the 4th step, this is because of a somewhat going transport, and that was gave a name as a transforming principle. And transforming principle, or it is DNA, or it is RNA, or it is protein, lipid or carbohydrate, it was not still cleared. So in a first test tube, the mixture of a heat killed a smooth strain and a wild rough strain plus a enzyme, that is a saccharolytic enzyme added, and this mixture is injected to mice. If that transforming principle is a carbohydrate, and because of a addition of a carbohydrate denaturing enzyme, the transforming principle also get denatured. So there was not a transformation. There was not a transformation. Then injection of a first tube is made to the mice, and mice get died. So the 3 scientists Avery, McCarty and McLeod concluded that the transforming principle was not a carbohydrate. In second step, mixture of 2 strain plus a lipase enzyme and injected to the mice. Mice also get killed, and they concluded that the transforming principle was not a lipid also. In the third step, a mixture of 2 strains, heat killed smooth plus a rough and a protease enzyme injected to mice, and mice get died. So the transforming principle was not protein. In fourth step, mixture of 2 strain plus a RNase enzyme that destroy a RNA and made an injection to the mice. Mice get died. Means from that 4 step, the transforming principle was not carbohydrate, it was not lipid, protein and RNA. But when he added enzyme DNase into a mixture of 2 strains and made an injection to the mice, mice remains alive, they does not get killed. Hence, because of a DNase enzyme, the DNase enzyme denatures or destroy a DNA. So there was no transformation, and there was no transformation of heat killed smooth strain, and so they does not become deadly. And the mice does not So there was no transformation because the DNA was added in a mixture of two strains and mice remains live. So it was clear that a transforming principle was not carbohydrate, lipid, protein, RNA, it was a DNA. After the last step, it was concluded DNA was a transforming factor, and it was confirmed after a experiment of Harshy and Chase. The Harshy and Chase were worked on a T4 bacteriophages. Bacteriophages are the viruses which cause a infection to the bacteria. Host for these bacteriophages is a E. coli. The genetic material of these viruses is a DNA. Bacteriophages are viruses that infect the bacteria, and the bacteriophage contains a DNA core and a protein coat. The Harshy and Chase make a use of two radioactives, that is a sulfur 35 and a phosphorus 32. The protein coat of bacteriophages were labeled with a sulfur 35. In a image we can look for the bacteriophages and its protein coat is labeled with a sulfur 35 as it in a pink color. In a second, he labeled a DNA of a bacteriophages with a phosphorus 320 or 32 in a green color. It is for just your illustration. Harshy and Chase, these two scientists mixed this radioactively labeled viruses with a bacteria, that is a E. coli. The viruses or bacteriophages make an infection to a E. coli. After it, the mixture of virus and bacteria were mixed, and after mixing, he separated the mixture by a process of agitation. After centrifugation, the centrifugal mixture contains a pellet at the bottom, and a pellet contains a bacteria because of their high weight. In a first tube, where the scientists made a use of a sulfur 35, in a pellet there was not a presence of a sulfur 35, and in a test tube where the scientists made a use of phosphorus 32, in this tube at the pellet they measure a radioactivity in a pellet, and they found that the pellet is with a phosphorus 32, and after it they concluded that DNA is a genetic material, or it carries a genetic code. How did the DNA, how it stores information and how it duplicates itself? The scientist Linus Pauling in 1940, He discovered the alpha helical structure of a protein. In 1950, the Erwin Chargaff, he said that in a DNA there is a presence of an equal amount of adenine and thymine to the equal amount of adenine and thymine to the guanine and cytosine. The number of purine and number of pyrimidines in a DNA is same. It was given by a scientist Erwin Chargaff. In 1951, the scientists Franklin and Wilkins, they gave a X-ray diffraction image of a DNA. And in 1953, two scientists James Watson and Francis Crick gave a double-stranded helical model of a DNA. We know that the DNA is made up of a nucleotide, that is adenine, thymine, guanine and cytosine. This follows a Watson and Crick base pairing. Adenine bonds with a thymine with a 2 hydrogen bonds, guanine with a cytosine with a 3 hydrogen bonds. Here is the bonding in between adenine and thymine, guanine to a cytosine. Each nitrogen base pairs with a sugar and to the inorganic phosphate. Here is a polynuclear strand or one strand of a DNA. DNA is arranged in a double helix. Now about the DNA replication. Replication means to making an exact copy of itself, to making a number of copies of DNA called as DNA replication. And DNA replication is a semi-conservative. From one parental DNA there is a formation of two daughter DNAs. Out of these two daughter DNAs, each daughter DNA has a one strand of parent and one strand is new. So this is a semi-conservative mode of replication. So thank you very much. Once again I request you subscribe my channel, like the video and share it to your friends.